So the second half of getting to a protein is a process called translation. So we talked last time about the process of transcription, which takes us from DNA to RNA, our mRNA, messenger RNA, which is able to leave the cell's nucleus. Now we're going to talk about this second part, translation, where we take this mRNA transcript and make a polypeptide. Okay, so the definition for translation is the process of making a polypeptide protein using mRNA as our guide, as our template for making that protein. It's going to occur in the cytoplasm, so out in the cell. It's going to use our ribosomes to build those proteins. We'll take a look at how that works. So translation actually takes a lot of energy. The cell works really hard on this, and it's going to require a lot of different components. 3 of them are different types of RNA. So we've seen our mRNA, our messenger RNA already. That's coming out of the nucleus. It's basically our copy of DNA in RNA form. That's going to give us the code for how to build this protein. We're also going to need to use transfer RNA, which we'll talk about, but there's a special RNA that carries amino acids to the ribosome. It's called transfer RNA, tRNA for short. Now, our ribosomes themselves are actually made largely out of RNA. We're not going to talk about how ribosomes get made, but you should know that they are a type of RNA. So we'd call them rRNA, ribosomal RNA, if we were thinking about the types of RNA involved in this process. Okay, as we actually do translation, we start with our mRNA transcript. So that's the recorded part of our gene. We've cut out all the parts that we don't need during our processing. So we have now just our long string of letters. This is going to code for our amino acids in our polypeptide. So the bases in mRNA, so bases like adenine, cytosine, guanine, and uracil. Remember, mRNA doesn't have thymine. They're going to get read 3 letters at a time. So sometimes we say that we're reading RNA triplets. Specifically, if we're talking about a specific set of 3 bases, it's called a codon. Okay, so we're reading our mRNA codons. Our typical polypeptide is like a few hundred amino acids in length, so it gets pretty long. So we're going to read a lot of different codons. I'm not going to make you build a 100 amino acid polypeptide here, but you should know that they do get quite long. Okay, so when we start to make our polypeptide, the first thing we're going to do is look for our start codon, which is going to be AUG. When we want to finish making our polypeptide, we're going to stop making our polypeptide when we hit a stop codon, which are UAA, UAG, and UGA. We're actually going to look at a table of all the codons, which is what you're going to use in order to build these polypeptides. But first, I want to make sure that we understand something called the reading frame. Okay, so our mRNA is a long sequence of letters, right? Each letter is a base for our nucleotide, right? A nitrogenous base. Because we added caps and tails and stuff to our mRNA, and because sometimes breakage happens, you don't actually need to start reading an mRNA right at the beginning at the first letter. There's often going to be like a repetitive sequence there, or something that's not really necessary to make our functional polypeptide. We know that we need to actually start pulling together amino acids when we hit 3 letters in a row that we recognize as our start codon. Our start codon is AUG. So if, for example, we had this strand as our mRNA, and remember we have to go from the 5' to 3' direction. Actually, don't remember, I'm telling you, we're reading it from the 5' direction to the 3' direction. So if we have the strand AU, AA, GG, AGG, etc., we're skimming this until we find our start codon AUG. So we don't see A, U, and G in that order until we get to this part right here. Okay. Now we see AUG. So this is actually when we're going to start grouping our letters in sets of 3. So they're not like physically separated into sets of 3 in the mRNA. It's all dependent on when we find that start codon, and then everything after that start codon is read as a set of 3. So you have to sort of group them together in your mind. Here I've grouped them with parentheses. Okay, so here's our start codon AUG. So our next codon would be CAG, then CAG, GGC, UUU, ACC, and so on. And we would actually keep reading this until we hit one of our stop codons, which we would be able to find in our codon chart, which I'll make available to you. Okay. So we're going to be pulling together amino acids based on these codons. So this is what a codon chart looks like. So you can Google these. I'll give you one if you need it. But it's always the same. So what this is a table of the letters you would find on your mRNA transcript and what a group of 3 would mean, like what amino acid it codes for. It's structured for ease of reading. You see AUG, so you find the first letter on the left-hand side, right? So if we had ACU, right? Let's say, okay, so we have ACU, so we look for our first letter A here, okay? And then we need to find our second letter, AC. Everything starting with AC is here. Okay, THR. That would be our amino acid. Okay, so once you identify codons, you make sure you find the start codon first, then everything after it gets grouped in triplets, so our sets of 3, each of which is a codon. And then you refer to this codon chart, find those groups of 3, and find out which amino acid they code for. Okay. So that's how we read an mRNA, and this is basically what your cell is doing as well. So if we went back to this example, AUG actually always also brings over an amino acid MET. So that's actually on our chart. Here you can see it's our start codon. It's going to bring MET. Okay. Then our next codons, GLN, GLN, GLY, PHE, THR. Okay. So each of these little codons is going to bring over an amino acid. That's a sort of conceptual vision of how we're going to get from our mRNA to our protein, to our polypeptide. But we do want to look a little bit at the cellular mechanics of this. So the way this is happening is basically a ribosome is going to snap together around our strand of mRNA. It's going to slide down the mRNA. And our final type of RNA, our transfer RNA, our tRNA, it's going to act as a little car or a little boat, bringing over the right amino acids to the ribosome. Okay. And the ribosome is then going to do the job of actually linking together those amino acids, forming peptide bonds, which are a type of covalent bond that we find in proteins, to make our final polypeptide strand that can go on to become a big functional protein. So let's take a look at tRNA, that sort of car or boat that's going to ferry over amino acids. So tRNA looks kind of like a cross or a lowercase t, but what we're really focused on is the 2 opposite ends of our tRNA. So at one end, our tRNA is going to carry its amino acid. So the amino acid isn't part of the tRNA, but it binds on to this little sort of area where there isn't the RNA is folded around on itself. So there's a little area where the RNA is single-stranded, leaves a gap for the amino acid to bind to. So here our amino acid is binding on at one end of the tRNA. And at the opposite end of the tRNA, we have 3 letters on our RNA facing the outside. And that's the part that's going to match up to your mRNA. It's basically going to sort of magnet the tRNA to the mRNA, which is how we're going to make sure that we get the right amino acid in the right order. So in order to magnet onto the mRNA, which is our actual code, our tRNA is going to have 3 little letters facing the outside called the anticodon. So they're just going to be the opposite of the codon. Their job is to come over and be attracted to the mRNA. The point of that is really just to bring this amino acid along with them. So here we can see that the anticodon on this example tRNA is GGC. So that would match up to an mRNA of CCG. Okay, that would magnet it on. It would bring our amino acid with it. Okay, so just like there were stages to making our mRNA transcript from DNA when we were talking about transcription, there are also a couple stages for translation. They're pretty much named the same, but they look a little different because now we're at a ribosome out in the cytoplasm instead of in the nucleus using our RNA polymerase. Okay, so the stages of translation, the first stage is going to be called initiation. What happens is that our ribosome is actually a big piece called our large subunit and a little piece called our small subunit. So it has 2 halves that are kind of floating separately around for a little bit. When we want to start translation, they snap together into one ribosome. And they snap together with the mRNA and our first little tRNA with our first little amino acid stuck in the center. Okay, so here we can see, for example, our start codon, right, would be this green part. And this would be our MET, right, and our first tRNA. Okay, so it snaps together, forming this complex. So complex just means like a group of things working together. Okay, so the ribosome is then going to slide down your mRNA, sliding from the 5' end to the 3' end. Okay, and as it does that, it's going to be bringing in new tRNAs with new amino acids. So this part is called elongation because our polypeptide is getting longer. Okay, so our new tRNAs are coming in. They have their little anticodons, which magnet on to our mRNA transcript. They pass this little blue ball. This little blue ball is our amino acid. They pass it here, and as the ribosome slides, this little amino acid gets linked onto the growing polypeptide chain, and then our tRNA is going to come out the other side as the ribosome continues to slide. So this keeps going, keeps going, keeps going until we hit our stop codon. So here we see our stop codon in red on our mRNA. When we hit the stop codon, the ribosome falls apart. So the large subunit and small subunit break apart. Our tRNAs on the inside get let go, and so does our completed polypeptide. So this polypeptide chain that we were building because we want to make a protein gets freed into the cytoplasm when the ribosome lets go of everything it's holding onto. So at this point, we have our completed polypeptide and can start doing its folding thing, right? We remember that proteins have first fold into alpha helices, beta pleated sheets. That was our secondary structure. Then they had their tertiary structure where they ball together. And then maybe they work together with another polypeptide in a quaternary structure. If you want to remember about that stuff, go back to the proteins lecture. Okay. Final thing about ribosomes is that we can actually name those 3 parts of the ribosome that we were talking about. So the place where amino acids come into the ribosome, so our tRNA is carrying the amino acid into this first site, the amino acyl site, or the A site. Remember, the ribosome is moving this direction. So like the ribosome is moving to the right here. It's moving 5' to 3'. So the tRNA and the amino acid come into the A site here. That's our amino acid acyl site. And then the actual bonds between the different amino acids are going to form once the tRNA is in this middle site, the P site, peptidal site for peptide bonds. So you can see that's where we link on our little new amino acid here highlighted in yellow onto our polypeptide chain. And then when the tRNA hits the E site, the exit site, it falls out, makes room for the next tRNA. And that's it for translation. Now we have our protein.